Just gonna wait a few minutes for people to log on. Got one viewer. I think they count me as a viewer. All right. So, all right. Talking about two different types of transport. So both of these explain how things move across a cell membrane. So you have passive transport and you have active transport. The two types of passive transport are diffusion, or you might see simple diffusion, and facilitated diffusion. So both of these, what we're going to see, passive transport, two similarities that they do not require energy and that they follow the concentration gradients. So they do not require energy and they follow something known as the concentration gradients. Bless you. Okay. So, simple diffusion. Don't go crazy with the phospholipids, just draw 4. Okay. So if this is a cell membrane that continues on around a cell, okay, simple diffusion most often we're referring to the diffusion of carbon dioxide and oxygen, which usually is small, nonpolar molecules. Small, nonpolar molecules. And these simply can pass through the cell membrane, the phospholipid bilayer, without the need for a protein to facilitate it. So they're going to go in a particular direction. So carbon dioxide will diffuse in that direction because carbon dioxide is high on this side and low on this side. Right? So carbon dioxide will diffuse until equilibrium is reached. Right? So in this case, imagine that this carbon dioxide moves. Right? We'll have two carbon dioxides on both sides. So we have equilibrium. Oxygen will go the other direction because we have more oxygen on this side. Right? So diffusion just simply passes through the membrane. Okay? And we say that net diffusion occurs until equilibrium is reached. All that means is that there's diffusion always happening, but this net diffusion, this direction of diffusion, only happens until equilibrium is reached, equal size. Okay. So, an application, where do you think this is happening in our body? A whole lot. Where simple diffusion is happening a whole lot. What do you think, Helene? Our lungs, right? So there are particular cells in our alveoli, which are inside of our lungs. So this happens our lung cells? A lot. This is the whole basis of when we take a breath. The simple diffusions are happening. This is happening in all of our cells, though, as cellular respiration is taking place, but to highlight, happens in our lung cells a lot. Okay. Simple diffusion. Like I mentioned, concentration gradient, we're going from high concentration of oxygen or carbon dioxide to low concentration. That's all concentration gradient refers to, is this idea of high to low. Okay, questions so far? We good? Okay. All right, so facilitated diffusion. This is typically for large and or polar molecules. Right? So this allows large and or polar molecules to pass through the membrane. And it requires a transport protein. Transport protein. So, inside of the phospholipid bilayer, you have these transport proteins that are embedded in there. Just as a review, remember that you also have something else embedded in the membrane. Does anybody recall what's this molecule that helps stabilize the phospholipid bilayer? Cholesterol. So cholesterol, just as a reminder of things that are embedded. But these transport proteins, right here, they allow either large or polar molecules to pass through. So for example, we have glucose. Glucose is a large molecule. It's not polar, but it requires a transport protein to move through the membrane. And notice we're still going from high to low. High concentration at the bottom to low. Until equilibrium is reached. So this particular example is a glucose transporter. Very common transport protein. Okay, you also will see, so here's some examples, glucose transporter and ion channels are another example. All right, ions, let me add something here real quick. Large and or polar / charged. Right. So an ion, right, is a charged molecule, can't make its way through the nonpolar portion, so it needs a channel. If you're in my bio class last year, you remember one example of an ion channel, which is the chloride channel for cystic fibrosis. But an ion channel and glucose transporters. Okay. So, where do you think in the body a glucose transporter would be really important to have? Think about where your body needs to move glucose across the membrane. Okay. So the pancreas will often help signal cells to bring glucose in. Right? We need glucose for cellular respiration. That's where energy is stored. Right? But one of the most common places that we see glucose transporters are in intestinal cells, right? So intestinal cells are constantly passing glucose across their membrane as they absorb it from our food. But again, when I say these two, like the lung cells, intestinal cells, doesn't mean they're not found elsewhere. Those are just one highlight of where they're commonly found. Okay. Questions so far? So simple diffusion, facilitated diffusion. They both do not require energy, and they both follow the concentration gradients. So high concentration of glucose to low concentration. Glucose. Okay. So, the last one, we've got simple diffusion, facilitated diffusion. Active transport, the two things that we're going to remember here is that active transport requires energy, or ATP to be particular. And active transport goes against the concentration gradients. All right. So first of all, one thing we need to talk about. So energy, or ATP, which is charged by the mitochondria during cellular respiration. It's a form of chemical potential energy. It has a specific structure that we're actually going to take the time to draw because it's super important. You're going to see it a lot. So it's a big one. So we have a phosphate group. Connected to a phosphate group. Three phosphate groups. connected to, what is this right here? A sugar, right? This is a sugar. All right, we're halfway there. I'm kidding. That's it. That's the last part. Okay, so there is ATP, which stands for adenosine triphosphate. Super important molecule. We'll talk a lot about it during our next unit. Okay, a couple things to notice. I'll take this one away a second. What does that look like? What does that look like? It's a particular type of monomer. Nucleic acid, right? Phosphate, sugar, nitrogenous base, right? So, what's cool about this is that adenosine triphosphate shares its structure, aside from the extra phosphate groups, with the base adenine. Adenine connects with thymine, right? Adenosine triphosphate has a very similar structure, except it has a couple extra phosphates, right? All right. So, this phosphate group, this third phosphate group, we often refer to this as the first, the second, and the third. The third one, it stores all of this energy such that when this bond right here is broken, when that's broken by, let's see if we remember, this is a polymer of phosphate groups. What do you think is involved in breaking that polymer bond? Hydrolysis, right? So when it's broken, it releases energy. And that bond right there that I'm pointing out is broken. It releases tons of energy because that third phosphate doesn't want to remain there. It's unstable. So it breaks off and releases energy. I think that's a symbol for energy, right? Maybe that's Harry Potter. I don't know. Okay. So this is the major form of energy that the cell needs in order to actually function. So we'll get to this later, but cellular respiration, it actually charges the ATP molecule by forcing that third phosphate on there. It's like a charged battery, so that the cell, when it's ready to break it off, it can release the energy that's stored there. So we'll get to that a little bit later, but that third phosphate is where the energy is stored. Okay. So what does this have to do with active transport? So active transport, we've got our cell membrane. We also have proteins embedded for active transport. Hello. Thank you. Okay, so active transport. So what I've drawn are some sodium and some potassium ions. Potassium is the K. Okay. This is probably the most common active transport situation. So where we have this protein actually pushes sorry, I drew that wrong. I'm going to add some more potassiums down here. Okay, so sodium is being pushed against its concentration gradient, right? It's going from low concentration of sodium to high concentration of sodium. Potassium is going to go the other direction. Pushing sodium across its concentration gradients. Now, in order to do this, this requires ATP to essentially pump the molecules against the gradients. In the way that we usually represent that. ATP becomes ADP. What do you think ADP stands for? Right, adenosine diphosphates, because you've lost that third phosphate, right? So that process releases energy, allowing you to pump the ions across the gradient. All right. So we have ATP helping pump the molecules against the gradient. We have a protein that's helping to move these molecules across the membrane. Where this often occurs does anybody want to offer up? Where does this often occur? Often occurs in neurons. So neurons are pumping ions across their membranes to create something called a membrane potential, which we'll talk about later. But neurons are always doing this. Okay. Forgot what a neuron is. We're talking about brain cells. All right. So we have passive transport, active transport, ways of moving molecules across a membrane in certain directions. Okay. So the last thing I do want to talk about, this is technically active transport. Technically active transport. I'll spend a short moment talking about it. Is you have something called endocytosis, and exocytosis. If you need to turn over to the next page, you can. Just a quick note about these two. So endocytosis and exocytosis are two terms that you'll see. These two, they help move large molecules, often in large quantities, across a membrane. Okay, so if I have a cell, I'm going to draw just two basic things in the cell. So I've got my nucleus, and I have my Golgi apparatus. I'll just put Golgi. So my Golgi apparatus, which we know its job is to send things out of the cell. So what happens is that we have these little vesicles that pop off. The Golgi apparatus. And they actually fuse with the membrane. So what'll end up happening zoom in right there. So what ended up happening is that whatever is inside those little vesicles will be released out of the cell. Right? So it's like those little vesicles that are coming from the Golgi apparatus, they contain often proteins. They move to the cell membrane, and they actually fuse with the membrane and release the contents. Okay. We call that exocytosis. Exocytosis. The most common place that we see this are in synapses of neurons. So oftentimes these are little red dots are neurotransmitters that are being sent across a synapse. Synapse kind of looks like this. These are neurons. This middle part is a synapse, which basically allows neurotransmitters to be sent from one neuron to the other. That's how signaling happens across neurons. Okay. So if exocytosis is pushing things out of the cell, what do you think endocytosis is? Bringing things in, bringing things in. Hopefully this will look familiar to you. So what we have are the cell membranes. They kind of bend inwards, and they absorb what's outside of the cell. Where have we seen something like that before? Right, the endosymbiotic theory, right? So endosymbiotic theory is the endocytosis of a prokaryote. Yeah. Okay. Questions? So endo and exocytosis, they're technically forms of active transport, but all you need to know is that they allow large quantities or large molecules to move across. All right. I'm going to stop talking. What I'd like you to do, go ahead and take a 2-minute stretch break. We'll come back to the last part break. Yep.